All right, so let's open the floor for questions. Thank you very much to the speakers for presenting these spectacular results from the research.
Thank you very much for excellent talks. So Adriana, first question. I recognize that you don't deal with humans, you're dealing with animals. But I've got two patients who've got celiac disease and have got Wilson's. And I was very interested to see that you thought there may have been a relationship between celiac disease and Wilson's. Is it just fate that they've got Wilson's disease and celiac, or do you think the two might be related? And if so, how?
So, thank you for the question. In terms of the comorbidities, so that represent this group of the irritable bowel disease, so it's celiac, it's Crohn's disease, and it's the ulcerative colitis. So I don't know how they can be correlated, in the sense I don't know specifically how to explain when a patient has both. What it's known is that this is particularly more pronounced in children with pediatric patients with Wilson's disease. And when you go later, this is more Crohn's disease, so it's something more severe in adult patients. So currently in our lab with research, I'm now trying to put together this project very focused on inflammation, and that I hope that will help answer that question. And we also have now this collaboration with Piotr and Joanna in Poland to get samples from patients and try to understand. One thing we are trying to do now is to work with EPSCs, so these pluripotent stem cells that we get from the urine of patients, and that we can then differentiate in different types of cells, liver and intestine, from pediatric patients. And this could be a very nice tool then to answer the question if it's just fate in terms of celiac disease or no. So currently I don't have an answer, but hopefully we could.
I mean, just from my experience, what I did in those two patients was that I had a dilemma, because if I gave them treatment for the celiac disease, I was unsure about absorption of medication. So what I did was I made sure that the celiac disease was under control, re-biopsied them, and just made sure it was absolutely under control before I gave them oral chelation therapy. So I thought that'd be a good experience to share. No,
this is a very good experience to share, because we know that if the intestine is damaged, this can really influence liver inflammation, and this can trigger the liver to be damaged way earlier. That's why it's important when you try to control first the celiac disease and be sure that it's stable, then of course starting with the treatment. Because if the intestine is damaged, also the therapy will somehow be an extra burden for the intestine, because right now the penicillamine, trientine, zinc, they are all taken up via the gastrointestinal tract, and if this is damaged, I think we will have a worsening of the disease overall. That's why I think this part with the intestine, it's quite important.
Okay. Thank you. My second question then was Pasquale. That was a fantastic presentation. I really understood it. Well, so I think. But the challenges with gene editing and gene therapy, is there room for both in Wilson's disease, or do you think one is going to take over the other? I'm sorry, that's an unfair question.
No, that's a very good question, actually. So basically, we are now talking about two different approaches that are at two different stages of development. So gene replacement have reached the clinic, so it's under investigation. So we all hope that it will work pretty much. So if we have a very effective gene transfer using traditional gene therapy, that would be okay. Although we already know that this has been done only in adult patients, so pediatric patients have not been admitted to the investigation. So, and especially for this kind of patients, genome editing is an alternative because, you know, there we already know, especially if you are treating patients very early in their life, you already know that your gene therapy will be declining over time. That's pretty much what happens. So, in this case, you will go for genome editing, although the stage is still quite preclinical in this case.
Just building on that, just final question, just because I see over the last 12 months, the arena in rare liver disease is focusing more on gene editing than in gene therapy. Yeah. And it seems to be very exciting either way.
Yeah, of course, you know, there are many attractive features of the genome editing compared to the gene therapy. So gene therapy is like more a brute force approach. So you are just providing cells with an external DNA, with an external expression cassette, with the promoter that is the promoter of the endogenous gene. So you are basically overexpressing your gene, which is no longer regulated as in the physiology, normal physiology, and so on. So this is what these are the limitations of gene therapy. While genome editing, the promise of genome editing is to make precise modifications. So basically, you can even with base editors, for example, you can even modify the single base that is mutated. So it's the most precise approach that we could imagine. And there is also, I didn't have the time to talk about this, maybe next year if you want. So there is a lot of interesting stuff coming out also at the RNA level, for example. So I think that now the therapeutic platform that we could use to approach genetic diseases are really a lot. Most of them are in the preclinical, but some of them are already entering clinical investigation, even of these that have been developed like few years ago, like base editors, for example. They are now entering the clinic. So we'll see. For sure, theoretically, genome editing is more attractive because you have your gene in the same place, just with all the stuff and the location is the same as in the healthy individual, and you just make the small modification to make your gene work. So much better.
I'm sorry, I've got one more question.
No problem.
But if you had a crystal ball, and
I hope I have.
Yeah, if you which you might have, then what's your prediction for year 1, year 3, and year 5?
Oh, that's. So I have to say that this is not just. So I can reply on the scientific point of view, but the issue is that this is not just scientific. So and maybe I'm not the right person to talk about this, but we are now like a solid scientific foundation for this kind of therapies, and many groups and people, researchers all around the world are struggling to push this into the patients, into the clinic, to make translational studies and so on. But now the bottleneck is that clinical investigation is very expensive, and you cannot find easily people that want to invest in this, and also companies are not always interested in rare diseases. So I have to say that we are now facing the reality that we have the technical possibility to cure diseases, but we don't have the people putting money in to make this real. That's why it's quite hard to say what are we going to. Of course, we have 2 ongoing clinical trials for Wilson, and this is really something that is good for all the community. And so we hope that in 1-3 years that we have the results from these trials that maybe could open a new therapeutic opportunities for patients, but it's hard to say where is moving the field is moving. Yeah.
Congratulations for both presentations. Tengo dos preguntas.
I do have two questions for Pasquale. Two questions for Pasquale. One is a scientific question. The other one is not so scientific. It's very selfish. First one, I didn't understand well. That maybe that's my. Well, what's the difference between editing and the traditional system in terms of dilution? I mean, the ecological advantage of the healthy cell. See, this is just Darwinism. I survive because I'm better than my neighbor. This happens with the two systems. Why there is such a big difference between these two systems? Then the practical questions. I don't know whether you know or are aware about this study published on the most popular mutation in Spain, the methionine 648, I think. There was some exon skipping problem, not a change of amino acid. And well, if we have a double mode of action, it would be easier to correct through gene editing. And I know that some colleagues contacted them to collect these patients with this methionine 645 arginine. Are you aware of this mutation of that group that started and then did not continue?
Okay, so the first was about the difference between genome editing and gene therapy. Yes, it's almost let's say in situ Darwinism. So it's like when you have a group of two groups that have different characteristics, and they are basically dealing to survive and to expand. Those that have your correct ATP7B can deal better with copper, and so they could grow more. That's it. Yeah. So the issue is that when you use genome editing, your correction is just inside the genome. So this cannot be lost, because when the cell replicates, you got the replication of the DNA, and the alteration that you installed is there and it stays there. When you're using classic gene therapy, you have this piece of DNA that is just separated from your genome. So this does not replicate, does not bind to all the stuff that cells builds when it has to replicate and to separate, the two parts of the. And that we really don't know precisely the mechanism why we got this dilution, let's say, but likely is because of this piece of DNA lacks of these components. And in fact, one of the, let's say, the line of investigation to keep traditional classic gene therapy to last over the years is that we have to recognize these elements that are necessary for a piece of DNA to be conserved, let's say, by the cell when it replicates. It's not integrating. Yeah. Okay. And the second question, I'm not aware of this specific study, but of course, as I mentioned, basically with some tools that are now available for genome editing, we can modify more or less all the mutation. We can restore the correct DNA sequence in every mutation. One of the issues is the sustainability of this approach, because if you have a mutation-dependent approach based on a current regulatory framework, you have to develop a drug for each mutation. So this is a very expensive, as you can imagine. Nobody's going to do this. So what the field is, and all the people working on this is trying to get from basically from the regulatory authorities, is that we have like a plug and play authorization when let's say the platform in general is authorized because it has done all the study that you have to do when you want a new drug to get into the market. but you can change the sequence, for example, the target sequence, which is just a part of the whole. And this would allow for a mutation-specific approach. But if this is not, this will not be achieved. We will not. We'll have this approach just for very common mutations, but not for those that are less prevalent.
¿Alguna pregunta más en la sala?
Any other questions?
Yes. First, thank you very much for these wonderful, wonderful talks. I mean, I really enjoy both of them. So I first have a question for Adriana. I mean, you have shown that you have a lack of integrity in the intestine in these animal models. I wonder, and I already talked to you about this, but do you think that there are also issues with the microbiota of these patients or these animals that can also be associated with some of the pathology that you are observing?
So there are already some papers, recent papers that show changes in microbiota in patients. I had two references here in my talk, but there are more. In the animals, we didn't study yet the gut microbiota. That is what we want to do next. But we have colleagues in the US, Valentina Medici, and also, I'm not sure if Svetlana already did microbiota, but Valentina Medici did in the mice, in the ATP7B knockouts, and they have alterations in the gut microbiota, naming less overall variability.
Diversity?
Diversity, thank you. So the microbiota is less diverse, and there is more predominance of what they call not so good bacteria. So this is, actually we were preparing a manuscript together, but I don't know if it was already published, also in the question of the lipid metabolism, because these animals, both rats that we have in our lab and the mice, they also have several issues with lipid processing and lipid transport into the bloodstream. And what we cannot forget is that we have an issue with copper excretion through the bile in Wilson's, and this per se changes the bile, and the microbiota are very reliable on the bile components, especially the short-chain fatty acids. And from the moment that our bile is changed, that the lipid processing is changed, this will undoubtedly, my English today, will change the gut microbiota. And so we have this indication on patients, we have this indication on the mice. With our rats, we still don't have data for the microbiota, but this is planned as a next step. So as Pasquale said, maybe there will, in the next year, more development. So if you want us both to be back, I think it will be very, very nice. Thank you.
Okay, thank you. For Pasquale, I really hope, and I think the next medicine is based on gene editing for rare diseases, for sure. I mean, the issue I see is still the efficacy, no, and of course safety, no. But first, maybe efficacy, and you have shown that you have like 100% repopulation with the corrected hepatocytes, but this will be the case in patients. So you have a selective advantage in mice, but in patients, will you remove the treatment? So, because you need damage, you need liver proliferation for repopulation with corrected hepatocytes. So I'm thinking on how to translate your results to the clinic. No, how will be the protocol, and if the clinician will take the risk of removing the treatment. So I want to know your opinion.
Yeah, so of course this is a very severe model. The mouse is a very severe model. This specific mouse that we are using, we tested this approach also in another mouse that has a less severe disease, and the percentage of repopulation was lower. And I think that this, of course, will not happen at this degree of magnitude in the patients. We will not expect this. although we made an additional experiment in which we injected animals with the vector when they were under treatment with the penicillamine, and we do see some expansion even in this case. Of course, the relation of the expansion with the damage is clear, and so this is actually a very good topic to discuss with clinicians, as you see, because at some time you have to taper the chelation and try to make the genome editing working. The other way, as you were saying, is that to have more efficient genome editing. So at the very beginning, you already reach like a threshold of which the benefit is there, and then you have a slower growth of the edited clones over time without exposing patients at a risk. Yeah.
Thank you both very much for the presentations. I have a very short question about genetics. So for genetic replacement or genetic editing therapies, I understand usually this is done through injections. Is there also any other way to do, even if at reduced effectiveness, is there any other way like ingestion for this kind of treatment, or is it really only the way to inject it?
So for this kind of vectors, so viral vectors, AAV vectors, blood infusion is the way, especially if you have to reach the liver, because liver has huge blood supply, so everything that is in your blood goes through the liver many times, and so the best way to reach the liver is by the bloodstream. Although this is, of course, something that may, let's say, if there is a more confined way to deliver a gene therapy vector to the liver, I will be happy to do it, because when you inject systemically, like in the bloodstream, it goes to the liver highly efficiently, but it also goes somewhere else. So this may be associated with some risk, but at this moment, the infusion is the best way to perform it.
And a question for both, also following this point, would it be possible to also improve intestine cells, digestive cells, through genetic editing, also through ingestion of some treatment? Is this something that's possible?
No, not at the moment, because of course the GI, the gastrointestinal tract, is not a very friendly environment for a gene therapy vector, for genome editing machineries. This, of course, is something that, let's say, precludes many diseases to access to gene therapy. And another issue with the modification of and the therapy, gene-based therapies for GI tract, is that the cells in your intestine are replicating much faster than your liver cells. So you have to address the right type of cells with very high efficient to think about correction of to have a therapeutic benefit, and the area that you have to target is huge. Like the area of your intestine is like this room probably. So that's quite hard to think about at this moment at a gene therapy based approach for this kind of diseases. Yeah.
Thank you.
Por aquí.
It's a question for Pasquale. How far are we from reaching a solution for gene therapy or gene editing when would be your guess when patients will have the opportunity to have a cure on? What would be your guess? It's something about 5 years, 10 years?
Yeah. So just to make a
2
guess,
years.
like a crystal ball that Abu was mentioning. Yeah, I think that next 5 to 10 years will be quite crucial. So yeah, 10 years is a reasonable horizon. Time horizon, let's say.
And I have another question. How committed is like the scientific society with the disease, with our disease, with Wilson disease? Is this something that it's being on a priority because it's something that is reaching a favorable scenario, or this is my question.
So I've been working on rare diseases since I started the university studies. So I have to say that Wilson disease is not really like other rare diseases, because especially if you are dealing with the pathologist, it's a quite known disease. While if there are many clinicians that work on the liver that don't even remember some of the rare liver, very rare liver diseases. So it's a quite well investigated topic, both at the liver, but I guess also at the brain level, and is not so common. This means that many therapeutic tools that could be applied to many other diseases are first tested in Wilson, for example, in the liver, and some other disorders. So I think that there is quite an amount of attention on this disorder. Yeah.
Thank you.
I apologize with the pathologist in the room.
Muchas gracias por las.
Thank you very much. Thank you very much for your presentations. I have a question. What experience do you have with patients? And if there's research with patients, what's the status of this research? Because you've mentioned gene research, and this research is improving.
We know that this is improving models, animal models. We don't know yet if this is improving the disease in humans. That's why we have the clinical trials. So we have a small amount of patients that are, in a really, tightly regulated experimental setting. They are tested for the new therapy. So we don't know yet if the traditional gene therapy, classic gene therapy, will work in these patients. We will know in the next year, and all the other platform that I mentioned need to pass through this kind of process. And so we can try many different models, and we know that from our experience that even in the non-human primate, when you tested something in non-human primate, you have very encouraging results. This doesn't imply that translates with the same efficacy in the humans. So this is something we have to wait to know, and let's see. Yeah.
Okay, and this is a message for the rest of patients with Wilson's. They're trying out gene therapies in Spain. There's a hospital in Barcelona that is conducting a study, and they're looking for patients to enroll with Vector V801, and there are no volunteers. They need volunteers to enroll. So I'm making a call to patients with Wilson's who are overage. So do participate in clinical trials, please. Any further question in the room? All right, I think that there's no break, mind you. I'm telling you. You guys, we're going to continue, but let us put our hands together for our speakers, Pasquale, Adriana. Thank you for presenting your results.